Hello, we are back to discuss topics and concepts from Chapter 7, and we will complete the presentation with a little bit of information about the lab experiment that we will be conducting. The next major topic that we will talk about is called cell respiration, and please read Chapter 7 in your online textbook for more details. What is cellular respiration? You can see that I wrote some of the definition already for you, but let's write the entire definition for cell respiration down. Okay, so the definition of cell respiration is going to be the metabolic process by which cells convert chemical energy stored in organic molecules like glucose into ATP molecules. ATP is the cell's main energy currency. We briefly discussed ATP in Chapter 6, so please make sure you refer to Chapter 6 to understand what ATP is. The whole goal of the process of cellular respiration is to make ATP molecules for the cell. Let's draw a very basic diagram about the energy conversion that is going on during cellular respiration. We're going to start off with an organic molecule, like, for example, glucose. Please understand that various organic molecules can be broken down during cell respiration, but we start understanding cell respiration by looking specifically at the organic molecule called glucose. If you have a firm understanding about how glucose is metabolized during cell respiration, then that will allow you to understand how other organic molecules, such as other type of carbohydrates or proteins or lipids, are going to be metabolized during cell respiration. So we start off with a very high energy molecule called glucose. And what happens is that glucose is broken down in many steps, 15, 20 steps during the process of cell respiration. And the energy that was once trapped in the bonds that make up the glucose molecule are slowly transferred to the energy that is going to now be trapped in our ATP molecules. ATP molecules are going to be easily broken down by the cell to provide energy for various cell activities. So let's go ahead and make sure we note that the energy conversion that is happening during cellular respiration is that we are starting off with an organic molecule like glucose. It's a very high energy molecule. and that it is slowly broken down through many steps or many reactions, and the energy that was once trapped in the bonds that make up the glucose molecule will now be trapped in some specific bonds that make up the ATP molecules. The whole process is called cell respiration. Remember that ATP is the goal of cell respiration, or the end product or final product of cell respiration. And that ATP molecules can be used to provide energy for various cellular activities. One example of a cellular activity that would require the energy from breaking down ATP molecules will be muscle contractions. Think about your heart and how the muscles never stop contracting in your heart. And if they did, of course, that would not be good. Therefore, the muscle cells that make up the heart, which are called the cardiac muscle cells, have a very high demand for ATP molecules so that the muscles will keep contracting. Now let's get into some more of the details now that we talked about an overview of what was going on. It turns out that during the reactions that are included in cell respiration, there are going to be transfer of tiny energetic particles, which we call electrons. So do not forget that cell respiration is an energy conversion process. Now let's get into the details about how the energy is converted. So the energy is transferred during this process by the movement of high energy electrons. We'll put a little flash behind that electron to indicate it is a very high energy particle. During the process, we're going to have a couple of molecules that are specifically designated to collect these high energy electrons and deliver them to a specific substance. So basically, these substances are going to be taxi cabs for electrons. They're going to pick high energy electrons up, and then they will move the high energy electrons and drop them off so that the cell respiration process can continue. The transfer of hydrogens will indicate when electrons are transferred. Let's give an example of this, and it's going to be a very basic example of the process of transferring these high energy electrons from one substance to another substance. We'll start off with substance A. So substance A will have a high energy electron, and the only way we'll know that it has that high energy electron is the presence of a hydrogen. So here's the hydrogen, and that is going to be our indicator that this substance has a high energy electron. A reaction occurs where substance A is going to lose that high energy electron, therefore it will lose the hydrogen, and the hydrogen or the electron will be transferred to substance B. Since that electron is very high energy, that means that substance B is going to ultimately be energized. When a substance loses a high energy electron, and you can tell that it loses the electron because it loses a hydrogen. When that substance loses that high energy electron, we say that substance is oxidized. So substance A was oxidized during this reaction. The term oxidation means loss of a high energy electron. Let's repeat the same exact reaction and just notice another term. So remember, this is what we started off with. Let's talk about substance B. Substance B was reduced during this reaction. Reduced means that the substance gained a high energy electron. Because remember that when you see the hydrogen, that indicates the electron. Together, these reactions are called oxidation reduction reactions. Oxidation dash reduction reactions. They're also simply called redox reactions. This has all to do with how energy is transferred in living organisms. This is an absolutely critical process. Without redox reactions, cells would not be able to continue to live. Make sure you understand that if cells cannot make ATP, they will die. So therefore, redox reactions in which energy is transferred in order to make ATP molecules are absolutely necessary to continue life. Let's continue on page number 2. Let's talk about those electrons again, and we're going to now name 2 specific molecules that pick up high energy electrons and deliver them to specific areas and substances within the cell. So some substances found in cells are used to transfer electrons during these redox reactions. The goal is to transfer all of that energy to ATP. So remember, that is the goal of the whole process that we're talking about today. Without ATP molecules, the cell will not continue to live. So the first energy carrier molecule is called NAD+. This stands for nicotinamide adenine dinucleotide, but you can simply recognize the molecule as NAD+. Again, we're going to pick on our glucose molecule. We have to basically say, okay, our starting molecule is going to be our organic molecule that we want to break down in order to transfer the energy from that molecule to our ATP molecules. We just need to realize that breaking down glucose does not occur in one step. It is a very stepwise process. So we'll start off with glucose, which we know is high energy. During glucose metabolism, glucose is oxidized. So recall from the last page that oxidized means that glucose loses electrons. NAD plus is a molecule that is available to pick up the electrons that are lost when glucose is metabolized. When NAD plus picks up the electrons that are lost from glucose, it becomes NADH. This is the reduced form of the molecule. So we had one substance was oxidized, glucose was oxidized, and oxidized means it lost an electron, and that electron will then be captured by NAD plus to become NADH. So therefore, we have these reactions, oxidation and reduction, are constantly occurring back to back during energy conversion reactions. The next energy carrier molecule does pretty much the same thing, and it is very similar in nature to NAD. This one is called FAD, and it stands for flavin adenine dinucleotide. It is a very similar molecule. Both molecules are derivatives from vitamin B. Again, we'll start off with glucose, which we know is very high energy. And during glucose metabolism, glucose is oxidized. That means it loses electrons. This molecule is called an electron carrier because when it picks up the electrons that are lost from glucose, then it becomes FADH2. Please recall that all the energy that is now found in NADH and FADH2 will be all transferred to ATP during the process. Let's continue and do number 4. What are the steps of cell respiration? Well, this depends on the type of cell respiration. There are a couple different types of cell respiration. We're going to start off with something called aerobic cellular respiration. Again, the whole goal of all of these respiration pathways and activities is to make ATP. However, aerobic cell respiration is going to be a process that requires oxygen gas at some point. That's O2. During the process of aerobic cell respiration, glucose will be completely metabolized and many ATP molecules will be produced. The next type of cell respiration is going to be called anaerobic cell respiration. Anaerobic cell respiration. This is going to be a metabolic pathway that does not require oxygen gas. However, during this pathway, glucose will not be completely broken down, and there will also be a lot less ATP produced. Let's briefly discuss the types of anaerobic respiration that we see in living organisms. The first type is going to be called alcohol fermentation. Fermentation is a process that does not require oxygen, therefore, it falls into the category of anaerobic cell respiration. One of the types of fermentation is called alcohol or ethanol fermentation. This will occur in yeast cells. We will discuss more details about this process shortly. The second type of anaerobic respiration is going to be called lactic acid or lactate fermentation. This will happen in your skeletal muscle cells, and it will also happen in certain bacterial cells. So, yes, think about your skeletal muscle cells. Think about your biceps, triceps, hamstrings, quadriceps. There are over 700 muscles in the body. Okay, but when we're talking about specifically your skeletal muscle cells, okay, will undergo this anaerobic process called lactic acid fermentation. Certain bacterial cells will also undergo this process, and we will discuss a little bit more details later on in the presentation. Finally, the last type of anaerobic cell respiration would be seen in various organisms, for example, certain bacterial species, which use other substances like sulfur to carry out respiration. So therefore, these type of cells do not use oxygen to carry out respiration, but they use another element like sulfur or nitrogen to carry out cellular respiration. We're going to first pick on aerobic cell respiration and provide some details regarding that before we talk about the anaerobic cell respiration fermentation pathways. Please do not forget that aerobic cell respiration is a metabolic pathway. And a metabolic pathway is a series of many reactions that their goal is to make a final product. In this case, the final product is going to be ATP. So basically, aerobic cell respiration is going to be made of 15 or 20, 25 reactions that occur with the goal of making ATP. So ATP will be made a little bit along the way, but a lot of ATP is going to be made at the very end of this process. A lot of times metabolic pathways are broken apart into groups, which we call steps. For example, step one of aerobic cell respiration is going to be a series of reactions that we call glycolysis. So let's go ahead and write some details down about step one. So the whole process of cell respiration begins when the cell obtains an organic molecule that it can break down for energy. The primary source of energy is going to be the glucose molecule. So we're always going to start off with glucose is entering the cell. And this could be any cell type. This could be an animal cell, a plant cell, bacterial cell, etc. So here's glucose, okay, and it enters the cell. Does anybody remember the molecular formula for glucose? It is C6H12O6. How many carbon atoms does glucose contain? Well, it looks like it contains 6 carbon atoms. Let's look at the carbon atoms of glucose. So I'm just drawing a different picture of glucose molecules. So here's glucose. It contains 6 carbon atoms, and we'll keep track of what happens to the carbon atoms during the cell respiration process. So this is my glucose molecule containing 6 carbon atoms. During glycolysis, which is a series of at least 10 reactions, the glucose molecule is broken apart. The organic substances at the end of the process of glycolysis are going to be called pyruvate. So it no longer has the name glucose because we took the original glucose molecule and we're beginning the process of metabolizing or breaking down that molecule. Please do not forget that during this process, glucose is oxidized. Okay, and our cell has those energy carrier molecules that are readily available to pick up the electrons that are lost. I'm sorry, that's just supposed to say NADH, not NADH2. There are 2 NADH molecules made during the process of glycolysis. Glycolysis will occur in that weakly semi-fluid called the cytoplasm of the cell. 2 ATP molecule will also be produced for the cell, and 2 pyruvates will be produced at the end of glycolysis. So after glycolysis occurs in the cytoplasm of the cell will have gained two NADH molecules, two ATP molecules, and two pyruvate molecules will be left over. The pyruvates still have a lot of energy tied up in the molecules. During aerobic respiration, the pyruvates will move into the next step to be broken down even further. So the ATP that is produced right here is going to be made by a process called substrate level phosphorylation. So please make sure you review that in chapter 7. Let's proceed to step 2 during the process. Step 2 is going to be where we pick up where we ended off here. So we ended with two pyruvates. We still want to break these down. The cell is still interested in taking the pyruvate molecules and breaking them down completely to release the energy from those molecules. So we'll say the pyruvates during step 2 enter a structure called the mitochondria. To be converted to a molecule called acetyl-CoA. These molecules then enter a cycle of reactions called the Krebs cycle of reactions. This cycle of reactions is also called citric acid cycle. So the goal is to continue to metabolize the remaining parts of what was originally the glucose molecule. So we started off with glucose, but we started breaking it down. Our first step of breaking down glucose was called glycolysis, where we ended up with two pyruvates. The pyruvates then enter the mitochondria to be converted to another substance called acetyl-CoA. Acetyl-CoA molecules will enter into a cycle of reactions called Krebs cycle, which is also called citric acid cycle reactions, to be broken down completely. The goal is to break this glucose molecule down as much as we can to get all the energy from the original glucose molecule. So we're starting off with our pyruvates. So here's our pyruvate. This is only one pyruvate. So recall that we have two pyruvates, but I'm gonna draw one of them. and we convert that pyruvate to a molecule called acetyl-CoA. During this conversion, one of our electron carrier molecules is going to be working. And we also produce carbon dioxide. So this is happening in the mitochondria. This reaction is happening in the mitochondria. The acetyl-CoA will then enter a cycle of reactions called the citric acid cycle. Citric acid cycle of reactions. And the goal of the citric acid cycle is to take the acetyl-CoA and break it down completely to get all of the energy out of it. Along the way of the citric acid cycle, we have some carbon dioxide molecules produced. We have some energy carrier molecules produced. And we also end up with citric acid, which is an organic substance that we begin with at the same time as well. So please do not forget that these guys have energy. These are our electron carrier molecules. They have energy to offer. They have energy. We want to get the energy from our electron carrier molecules, and I'll show you how we do that on step 3. Let's quickly just make a little note here about what happens to this carbon dioxide. And some carbon dioxide produced. What is the cell going to do with this carbon dioxide? Animal cells will release the carbon dioxide. They're going to get rid of it because carbon dioxide is a waste to animal cells. So basically, cell respiration is going on in your cells right now. Carbon dioxide is being produced. It is waste. And our body is getting rid of it by breathing it out. So first it diffuses out of the cell, then it moves into our blood, and as our blood passes through the air sacs of our lungs, carbon dioxide is released out because it is a waste for our type of cell. However, plant cells actually like carbon dioxide. So plant cells will keep carbon dioxide and use it for photosynthesis. All right, let's move on to the next page. Remember that, recall from the last page that we had some energy now tied up in NADH and FADH2. Okay, so we had this energy from steps 1 and step 2. We had NADH and FADH2, which are energized electron carrier molecules. But remember, we want that energy to be transferred to ATP. So step 3 is all about transferring that energy to ATP molecules so that the cell builds its supply of ATP and then uses ATP as the cell needs activities to be completed. So NADH and FADH2 will transfer their high energy electrons to a system of proteins called the electron transport chain. It's abbreviated ETC. So here's the ETC. ETC is made of 5 proteins that are going to be found in the mitochondria. So let's look at this picture. This picture is showing a cell structure in an animal cell or plant cell called mitochondria. Please review chapter 4 if you need to review the cell structures that we have went over previously. The inner portion of the mitochondria is called the mitochondrial matrix. The mitochondria is made of 2 membranes, the outer membrane and the inner membrane. The electron transport chain is going to be a system of proteins embedded in the inner membrane of the mitochondria. So let's label this the ETC, which is the electron transport chain. So we see here that NADH and FADH transfer or give those high energy electrons to the ETC. So let's go ahead and write that in. So NADH will give its electrons to the ETC, and it will end up being NAD+. So it lost a hydrogen. That hydrogen moves to this space right here. This space is basically the space between the outer and inner membranes of the mitochondria. We have the same thing going on over here. The FADH2 will deliver its electrons to the electron transport chain, and it will then become FAD. So it lost the hydrogens. And those hydrogens are going to be sent to this area. So now we have a high concentration of hydrogen ions in this space. The space is going to be called the intermembrane space, which is the area between the outer membrane and the inner membrane. Let's go back to these electrons. So we have these electrons that are basically being moved from substance to substance within the chain. So movement of electrons through the ETC generates a gradient of hydrogen ions. What do we mean by that term gradient? That means one area has a high concentration and the matrix has a low concentration of hydrogen ions. And a gradient is anytime you have a difference in the amount of substances between two different areas or regions. So then what happens is we're going to let those hydrogen ions flow back from the space into the matrix. However, the hydrogen ions have a little positive charge, which you can see right here. And anything that is charged is not going to pass through a membrane very easily because membranes are kind of oily and oils don't like charged substances. So the hydrogen ions will move through a structure that is embedded in the inner membrane, and this structure is going to be called ATP synthase. The diffusion of hydrogen ions through ATP synthase is going to be called chemiosmosis. Okay, so what happens is the diffusion of hydrogen ions through this weird structure called ATP synthase is going to result in a process called oxidative phosphorylation at the site of ATP synthase. And it looks like this. Oxidative phosphorylation is when we have the low energy ADP molecules and we connect a phosphate to that to become our high energy ATP molecules. Please review the terms chemiosmosis and oxidative phosphorylation, as well as ATP synthase. I'll give you a moment to review the diagram and write it down. Okay, let's continue on and now talk about the overall equation for aerobic cell respiration. Oops, sorry. Okay, so this is something that you'll want to make sure you understand, and I also gave this to you in a handout. The overall equation for cell respiration is going to be glucose plus oxygen, and we have cell respiration, ends up producing carbon dioxide plus water plus ATP. Okay, so let's go ahead and add in the molecular formulas for each. So glucose is C6H12O6, oxygen is O2, carbon dioxide is CO2, and then we have plus ATP. So I'm just rewriting the same equation, just adding the molecular formulas. Additionally, let's go ahead and balance the equation. This means the number of atoms are the same on both sides. This is the reactant side, and then these guys are the products. Remember that heat is always given off. Okay, so a balanced equation means that you have the same number of atoms of each element on the reactant and product side. For example, on this side, there are a total of 6 carbon atoms, whereas right here you see C6, there's also a total of 6 carbon atoms on the reactant side. So when you study the equations for these pathways, make sure that you study the balanced equation. For example, I could say during the process of cell respiration, when one molecule of glucose is metabolized or broken down during cell respiration, how many molecules of carbon dioxide are produced? Well, it looks like there's 6 molecules of carbon dioxide produced. And if you wanted to know where are the carbon dioxide molecules produced, then you could go and review the steps of aerobic cell respiration. Let's look at this part right here, oxygen. We didn't mention oxygen anywhere in this process. So let's go back up to our picture and talk about oxygen. So here are the electrons. The electrons move through the electron transport chain. And as the electrons move through the electron transport chain, they end up losing energy. So the electrons at the very end of this process are very low energy electrons. Well, turns out oxygen, okay, loves electrons, and oxygen will take those electrons even if the electrons are very low energy out of the chain. Therefore, oxygen is what we call the final electron acceptor. During the process of cell respiration. That is going to be the role of oxygen gas. That is why we need to breathe oxygen gas in. If we have no oxygen available, then the electrons will accumulate in the electron transport chain and block the entry of new high energy electrons into the electron transport chain. Therefore, electrons are constantly being sent into the chain, okay, and move from substance to substance and then are released out of the chain. However, the electrons are very low energy at the end of the chain, and only a select few substances will accept those low energy electrons. And in this case, during aerobic cell respiration, that substance that accepts the final electrons is going to be the oxygen gas molecules. Okay, let's move on to the next page. And let's talk about our fermentation. So recall that we talked a little bit about anaerobic cell respiration. We said there were 2 types of fermentation that fell into this category called anaerobic cell respiration. The first type of fermentation is going to be called alcohol or ethanol fermentation. This is a lot simpler compared to aerobic cell respiration. So let's go ahead and look at what happens during the process of ethanol fermentation. So again, ethanol fermentation is going to happen in yeast cells, and it is the process by which yeast cells produce various alcohols, beer, wine, champagne, hard liquors, etc. So let's start off with glucose. Glucose enters the cell. In this case, it's going to be a yeast cell. Here's glucose. Remember, glucose is made of 6 carbon atoms. The first thing that is going to happen is glucose is going to be broken down by glycolysis. This is the same procedure as we have already discussed. 2 pyruvate molecules are going to be produced. During the process of glycolysis, NAD plus picked up an electron and became an energized electron carrier, and ATP, 2 ATP molecules were produced during glycolysis. This is a review of what we have already discussed. If the yeast cell is in an anaerobic environment, that means no oxygen. That means the yeast cell will not proceed to break down the pyruvates by the steps of aerobic cell respiration. Pyruvate instead will be converted to ethanol during a process called alcohol fermentation. And during the process of fermentation, NADH will give the electron back and then become NAD plus. Notice that the process of fermentation does not generate any ATP. It ends up producing alcohol and carbon dioxide gas. The problem is that if the yeast cell is in an anaerobic environment and there is no oxygen available, then the yeast cell will not be able to undergo the aerobic cell respiration pathway to make ATP. Therefore, the yeast cell is going to rely on glycolysis to make ATP instead of aerobic respiration. So in order for glycolysis to continue, we need to make sure that we have a supply of NAD plus to keep the glycolytic reactions occurring. All right, the second type of fermentation is going to be very similar, except a different substance is going to be made. And it's going to be called lactic acid fermentation, or lactate fermentation. This is going to occur in skeletal muscle cells and some bacterial cells. So again, glucose will enter the cell. Yay. The cell loves when glucose enters. So here's glucose. All right, don't forget glucose is C6H12O6, and here are the 6 carbon atoms of glucose. We're just drawing a very basic diagram of glucose to show what is happening during this process. Again, glycolysis will occur. NADH and ATP will also be produced. So what happens is that the cell says, oh no, I'm running low on the supply of my NAD+. And the only way that I can keep glycolysis going and make ATP is if I regenerate my supply of NAD+. Okay, again, these cells are going to be in an anaerobic environment. So this is dependent on the type of environment that these cells are exposed to. So if glucose is broken down during glycolysis in the cell, like the skeletal muscle cell, for example, a bicep cell or a tricep cell or quadricep or hamstring, just think about the muscles of your body. So it's one of your cells, or certain types of bacterial cells don't have oxygen in their environment. They're in an anaerobic environment. They're going to take the pyruvates and convert that to a substance called lactic acid. And that conversion is going to allow them to take their excessive NADH, which is accumulating, and drop off that electron and regenerate their supply of NAD+. So the reason they need to do that is so that they can continue the glycolysis reactions to keep making ATP for the cell, because without ATP being produced, the cell will die. That says fermentation. NAD plus is regenerated during fermentation. So that's the whole reason that fermentation occurs. In this case, lactic acid is pretty much a waste product. For example, if lactic acid accumulates in your skeletal muscle cells, it could lead to your muscle cells becoming sore. And the reason is because lactic acid, it basically is going to decrease the pH in that area of your muscles, and that actually triggers some pain receptors when that detection of the change in the pH is picked up. All right. Okay, so a little overall recap plus a couple of new terms down here. Both skeletal muscle cells and yeast cells can switch between aerobic and anaerobic pathways. So let's fill that in. They can switch between aerobic and anaerobic pathways, and this is going to be depending on if oxygen is available or not. Aerobic pathways that basically utilize aerobic cell respiration is more efficient for cells. And let me tell you why. So a more energy efficient. So aerobic cell respiration is basically going to be what the cell would prefer to do. It is more efficient for cells compared to anaerobic pathways. The reason is because anaerobic pathways, and think about fermentation, produce a lot less ATP for the cell, and they also make more waste for the cell that the cell has to then deal with and either try to get rid of or whatnot. Various type of microbes can switch between aerobic and anaerobic pathways, and these type of microbes are called facultative. That just means they can switch between aerobic cell respiration and fermentation. Okay, cells that require oxygen, meaning they cannot live without oxygen, are going to be called obligate aerobes. For example, certain bacterial cells that absolutely need oxygen, meaning that if you put them in an anaerobic environment, they are going to die. They don't have the capability to switch to anaerobic pathways. So these type of cells that absolutely require oxygen are called obligate aerobes. Almost done here. All right, let's do a quick comparison of aerobic cell respiration compared to anaerobic cell respiration pathways. So here's aerobic, and here is anaerobic. And don't forget that anaerobic is fermentation. So fermentation is a type of pathway that falls under the anaerobic group. During aerobic pathways, or during aerobic cell respiration, glucose is metabolized completely. But during anaerobic, glucose is not metabolized completely. This means that the cell is not able to break down that original glucose molecule to obtain all the energy that it can from the original glucose molecule. So in essence, it's a little wasteful for the cell. During aerobic pathways, oxygen is required. So yes, oxygen is needed, but over here, oxygen is not needed. So for example, in your skeletal muscle cell, if you're doing a very hard workout, sometimes your skeletal muscles become a little depleted of oxygen. You just can't get the oxygen to your biceps, your triceps, your quads, your hamstrings fast enough. So your cells in those areas may have to temporarily switch to anaerobic mechanisms, but then once you start breathing and getting oxygen in at a regular rate, they will switch back to the aerobic mechanisms because aerobic pathways are a lot more efficient for the cell. How much ATP is produced? I've seen various numbers, but 36 to 38 ATP per glucose molecule. That's a lot. Over here during anaerobic, only 2 ATP per glucose molecule. So that means when one glucose is broken down during aerobic cell respiration, the cell gains 36 to 38 ATP molecules. That's a lot compared to over here. When the cell uses anaerobic pathways such as fermentation, the cell only gains 2 ATPs, but some is better than nothing. All right, so we have a little on your own activity, so make sure to use your resources. But in lab, how can we determine the rate of cell respiration? We are going to be doing a cell respiration lab. We are going to use plant cells to do cell respiration. We're going to use garden peas. So basically, if we want to determine, hey, how much is the cell going through respiration, we could look at a few different things. Okay, we would basically go back to the original overall equation, and we could look at how much of the products are produced, or we can look at how much reactant is used up. So for example, remember that aerobic cell respiration is glucose plus oxygen equals carbon dioxide plus water plus ATP, right? I'm just rewriting the overall equation for aerobic cell respiration. Well, we could say, how much of this was produced? That would give us an idea about the rate of cell respiration that the cells were going through. If there was more carbon dioxide produced, then we'd say, okay, the rate of aerobic cell respiration was higher. We could also look at how much was used. If a cell used more oxygen, okay, we were able to have a device that measured how much oxygen that the cells used during aerobic cell respiration. If a cell used more oxygen, then we would say the rate of cell respiration was increased. So that is what we are going to be doing in our lab, and I will explain that during our lab class.